My name is Michael Skibstead. As long as I can remember, I've been a naturalist at heart. I spent a lot of my early years outside exploring and in nature. I sort of developed a really early kind of passion for nature and observation. In second grade, I developed this passion for reading. And I was in the car at a gas station when I realized sort of my passion for nature and reading could coalesce. And they did. I picked up this little book that was on the floor called Turtles. And I started reading through it. I was sort of looking at the pictures and reading through the caption. I realized immediately, this group of organisms is so unique. Morphologically, they're so dissimilar to humans. And as a second grader, this was something kind of fascinating realization. There was something so kind of foreign to us and diverse in, form and function. What other animals they were in the ocean, they were on land, they were in water, and every sort of ecosystem and environment in between. And I also also realized that there were sort of a diverse physiological functions these animals have that were extremely different than our own as humans, and even different than all other tetrapods, and there's not anything really that's even remotely similar. This was based on sort of looking at pictures of turtles that were brumating in the book, and as my passion got kind of developed more, and I started reading research papers and reaching out to authors, I started doing this in middle school, and I got involved with a lot of other field work that I haven't even touched upon in this work because it's sort of tangential. But I've done work in Texas. I'm actually doing some population modeling with open Jolly Sieber models right now that I haven't even sort of touched upon yet, but I'm helping with that. I've done field work with telemetry projects in Utah and Arizona. I presented at over 7 different conferences on various topics, from turtle spring ecology to my work in Madagascar that I've done with conservation. I've really dedicated my life to pursuing researching turtles for the sake of turtles. But I've realized, as my passion has developed more, that there's sort of 3 prongs to where turtles can take us and what we can learn from them. And overall, I think they kind of coalesce to make something that is really, I think, monumental, and there's a lot of untapped potential. There's the first thing is sort of the pursuing knowledge for turtles for the sake of turtles in terms of systematics, taxonomy, the population genetics and dynamics of turtles, and, learning about them for the sake of increasing our knowledge of turtles and herpetology. There's sort of a second discipline that is learning about turtles and their impacts within the environment. So this falls into like understanding population parameters and looking at what roles do turtles play as umbrella species, ecosystem engineers, keystone species, and how does this work within the ecosystem as a whole? Because biomass estimates for turtles are among the most for vertebrates. There's almost 900 kilograms per hectare slider biomass at a site in South Carolina, which is amazing. And they're having a huge impact on that sort of metric for their impact that they have in the ecosystem. And when we understand how they work within the ecosystem, we can better understand the ecosystem as a whole. It's really just sort of this amalgamation of processes. And these processes sustain our lives as humans. They also sustain us psychologically. And understanding how it works helps us better kind of, conserve ecosystem functionality. And so I want to understand, that's kind of the second discipline, understanding turtles for the sake of understanding the ecosystem for the sake of humanity. And then there's sort of this third approach, and I'll be talking about the ecological approach, but the third approach is, looking at turtles for sort of interdisciplinary connections. And as I've mentioned, the turtles span this kind of wide range of physiology that's super foreign to us, from their anoxia tolerance. They can literally extract oxygen cutaneously from water. They can survive on anaerobic glycogenesis for months on end. And how they do these things, I mean, it's incredible. We could learn a lot in terms of that. In biomedical and oncology, there's gene duplications of tumor suppressors that we could learn about for cancer research. And even in biofuel generation, there's so many things we can take away. I'll be focusing more on the ecology, but my specific research that I conducted with Dr. Greg Paul at the Los Angeles Natural History Museum, we wanted to investigate sort of the impacts that the non-native red-eared slider, which is a species of turtle that's listed as one of the top most kind of introduced turtles all over the world. And we wanted to investigate the impact that these non-native turtles have on California. One of California's only native turtles, the southwestern pond turtle. And we want to understand, within a sort of heterogeneous creek system, how are these species using habitat? Is there a difference in their habitat usage? And what does this mean? We also wanted to look at the southwestern pond turtles. How is habitat alteration affecting where we're seeing them? So we worked within this system that had sort of a lot of urban influence, but in areas was still natural. Now the question sort of arose from Greg worked about 20 years ago at the UC Davis Arboretum. There was one study on this in a very different system than where I conducted my research, but they looked at pond turtles and slider interactions and how they were basking, and they were looking at sort of, in invasive species ecology, there is sort of this assumption that it's an introduced animal, it must be bad, it must have negative effects, but realistically, what a niche is a very complex, the environment acting on an organism, the organism acting on the environment. And so there is potential that certain not introduced turtles could potentially not have negative effects when introduced, but we wanted to really quantify this. So we originally set out to do something with ecological niche modeling through a computer program, but could not get the permits from the park due to COVID and kind of the backlog there. But we kind of readjusted and we took an observational approach to this. And, we spent 6 months in the field. Surveys took approximately 5 to 7 hours. We did over a month of surveys. Got about 800 turtle observations of both species combined. And then we analyzed these. We grouped them in terms of, we had different basking categories turtles could be in. We would observe turtles with binoculars and categorize what species we saw, where they were, at 10 designated observation sites that sort of spanned the availability of altered to natural habitats. And we analyzed all of this, and our major takeaways sort of were that the pond turtles and sliders, where they're basking, they typically use different basking sort of substrates out of the 6 different designations we have within the river. And this was sort of interesting to see because Peter Lindeman, who's done work with map turtles, sort of showed that where turtles bask tends to be close to where turtles are in the water column and their home range. And so there is, the evidence there is just that these turtles are using different habitats to bask in, that we can't necessarily say for certain, but we can extrapolate that they're kind of separate within the water. We very infrequently saw both species basking together. There was one observation over 6 months. So they are partitioned in terms of where they are in the river when they're basking. The second major takeaway was that most of our pond turtle observations occurred at sites that were less altered overall, and basking substrate type observationally, deadwood, where pond turtles were frequently seen on, was typically associated with quieter habitats, more vegetation coverage, and pond turtles were less frequently found at higher noise levels, anthropogenic noise levels, whereas sliders were actually found at higher noise levels. And so it sort of showed that pond turtles were more congregated in terms of our observations of them in sites that were less altered by humans, and sliders were more kind of distributed evenly among the system. So these are kind of the major takeaways, and some of the forefathers of pond turtle biology, such as Bruce Bury, Robert Goodman, Dan Holland, all sort of pointed out, starting in the 1960s to 1990s, that pond turtles typically are kind of more rare at sites that are altered and reproductively not typically viable. So we think, based on these sort of the conclusion observation we made, that the first thing is that it's hard to say. Based on our research, it doesn't appear that sliders and pond turtles are really competing, at least for basking sites. There's more to be looked at here, though, in terms of in the water column, how are they interacting, in terms of for food and such, but it doesn't appear that that's the case. The second interesting takeaway is that pond turtles are sort of concentrated in the areas, based on our basking observations, that are less altered. And so that's sort of something interesting. It confirms those ideas from the past, and I'm currently working with restoration committees and targeting the area that's less altered for restoration of nesting sites for the pond turtles. And I'm also currently working with OC Parks on introducing deadwood, the pond turtle preferred basking substrate, to see how that affects where the pond turtles are distributed. Ultimately, this work helps us sort of better understand the ecosystem, and this is within a natural area, and so understanding this natural area and the species within it ultimately makes our experience sort of better and a healthier sort of population. So the Davidson Fellowship, I'm very sort of grateful for this opportunity, and thank you.